Hello, hi everyone. Am I clearly audible and must be visible? Hi everyone, my name is Anjali Ahuja and support. I think I'm clearly audible and visible. Can you confirm me in the chat? I think I am. I just checked it. Okay, so students who are hearing me for the first time, my name is Anjali Ahuja. I am the biology educator here at askIITians. And today we are going to discuss very important topic. We will be learning key concept of 12th class because in just few days, you people are going to have your biology exam, which already is on your head. And yeah, so I think we will be starting the session and I'm watching on the other screen because there's another screen I'm looking at. So I can see that if there is some student who have any question, anything, please let me know. If you have any query in the chat, you can put. Okay. And please ignore the background if you hear it. I'm going to share my screen and I'm going to start the lecture. Let me repeat the topic of today's session. I already have added some topics. Wait. Okay. Okay, so the topic that we are going to discuss today for the session is Foundation of Biology, Key Concept of Grade 12. So in short, in your grade 12, whatever topics which are very important for the session, we are going to learn that. I'm going to share my screen and the session is already there. My OneNote is visible to all. I hope it is. Okay, so one student named Aniket has joined the session. Hi Aniket, how are you? Aniket, am I clearly audible and visible to you, beta? You can keep your camera on if you wish to. Okay, and Aniket, I just want to know you are from which batch? I think I've interacted with you earlier, maybe in some other session, and there is other student also joining the session. The topic is already written on the screen, and I've added some important topics there, and we will be learning. I'll be taking your doubts also. As you see, foundation of biology, key concept of class 12th. First of all, onto the screen, you are able to see important topics, which for sure can be there in your paper. After that, out of this, I will be picking some important topic and I will discuss it. And if I get to have some question also, I have that question, by the way, we will practice that. But that depends what do you want? Because I'm going to take some important topic right now. After that, you're going to tell me. Okay, so here you see topic chapter wise are added actually. As you see, the first is reproduction in organism, where mode of asexual reproduction is quite an important topic. After that, from the second chapter, sexual reproduction in flowering plant, pre-fertilization event, inbreeding depression, and double fertilization. Very important. Matlab, on inbreeding depression, I'm going to put 5 star on that. So much important topic. After that, from human reproduction, gametogenesis, which obviously is a type of meiosis that you people need to learn. After that, fertilization and event of pregnancy. Aniket, you're hearing me, right? One student also joined, but yeah, there is some connection issue. Okay, and Aniket, till the time, if there are some doubts also now, because you already know it is so much critical time. So if you have any doubt, put it in the chat. I'll teach you that. Okay, now next, as you see from reproductive health, you must learn birth control methods, STDs and technique to treat infertility. मतलब ये जो technique वाला question है ना, on to this there are 100% chance that you will get MCQ also. Subjective तो there can be, but multiple choice question for sure, because I think every year I have seen a question on that. Gift, Zift, IVF. It's like they give a question that they will give you a scenario and then they will ask that what would a doctor recommend, something like that. After that, next is principle of inheritance and variation, which is genetics, which include Mendel's laws, post-Mendelian genetics, chromosomal disorder, and pedigree analysis. And even there is one more topic also, post-Mendelian genetic chromosomal disorder. Yeah, there is parallelism. You remember that what is the parallelism between chromosomal theory and gene theory? 3 marks ka question can come. So, parallelism between gene and chromosome theory. Guys, I'm telling you that you people have to do this topic. Out of this, if you feel that you do not know any topic and you're still struggling with that, that is the first thing you should do, because you have exams on your head. Okay. After that, from molecular basis, structure of DNA, experiment of Griffith, Hershey and Chase, all the experiment. Matlab, in short, experiment to prove DNA as the genetic material and how conservation, how DNA replication happened, which is semi-conservative method. That topic is also very important. Replication, transcription, translation, and lac operon. And out of this, lac operon say, matlab randomly, I'm reminding that from biotechnology chapter, insertional inactivation, because lac Z is there na. So that topic is very, 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 like 5 times very important. So I hope you understood. And Aniket, I'm requesting ki if you have any doubt, na, you can tell me, ma'am, this is the topic I don't know, and you can repeat that. I have full time to teach you. After that, next is this evolution from that organ, homologous, analogous ka to examples. Matlab, I will just write that you need to learn all NCERT example. Like in NCERT, na, they give it random. Some topics are given at the starting, and then some topics are given in between the topics, something like that. But you need to learn all the examples. Adaptive radiation, Hardy-Weinberg principle. You're going to have a numerical on this one. Aniket, have you practiced numerical on Hardy-Weinberg? Nahi? Are you serious? You have not practiced. Do you know the equation of Hardy-Weinberg? You're going to have a question on that. I think that is the topic. I'm going to highlight. Wait. Let's do one thing. I am going to tell you about Hardy-Weinberg principle, and we will do 1-2 numerical on that. After that, Hershey Chase. Do you have any problem from this one? Lac operon aata hai? You know lac operon? It is also important. Click confirm me, please. I'm going to move forward till the time. Okay, next is human health and disease. So from that life cycle of Plasmodium, this is very important. Life cycle ka to ye laga lo that this is the diagram you have to do, bache, because there is a diagram. You remember in life cycle of Plasmodium, and otherwise also they say that the life cycle is diagenetic. Ma'am, I'll be rejoining. Okay, no problem. Diagenetic means that any pathogen which is infecting any animal, it is actually living in two hosts. Like if I talk about Plasmodium, this is living in the body of mosquito also and in human also. So that is diagenetic. That means two hosts. And so life cycle mean half of the life cycle you have to draw in human, half of the life cycle you have to draw in like animal, insect, or whoever is the vector. So this causative agent pe to match the column type of question. That's my favorite type of question. Match the column. And their mode of transmission, difference between innate and adaptive immunity. AIDS ka to chalo easy peasy. Drug abuse mein all the drugs. There is a NCERT ka page also I can show you. Right now I will show you that. Just a second. So if you are going on to the chapter, where is health and disease there? And let me open the NCERT. So if you are here looking at the NCERT, I might have written something onto this, but there is a paragraph where all the drugs are mentioned, structure is given, important from where these are extracted and all that. So much important. Wait a second, this is also important. Anyways, I will not break the sequence. Let it open, give it few seconds. Yes. You have to learn each and every line and paragraph of drugs and alcohol abuse. This one. So much important. Matlab, yahan pe whatever I have highlighted, na, you can take a screenshot, guys. And you can save it, because whatever I already have added, I was teaching this in the class and I've already added a star on this, that this is the name of the drug and this is where it is extracted from. And whatever I have highlighted, questions already has come in multiple formats from this information. In NEET paper also and in board also. Right now we will talk about boards. And as you see, they have given cannabinoid molecule structure. This already has come in paper. Identify which one of the following. So that's what. Chalo, anyways, I would come back here onto the board PYQ. Okay. And next topic, strategy is deleted. So we will not talk about it. Microbes in human welfare, microbe and their product, sewage treatment, that is easy peasy. मतलब यहाँ पे microbes in their product में जैसे from which one of the following this type of cheese is formed, this type of propionibacterium, Shermanii and all that. This is very important. And then lactic acid from which bacteria. You're getting my point. It is like they'll be talking about the product and they will ask about the microbes from there where it is extracted. Next is biotechnology, restriction enzyme and the discovery. Guys, I want to tell you one thing, like whenever any unit starts, matlab there are 5 unit in 11th class, 5 unit in 12th class. After, like the first chapter of every unit has a paragraph in NCERT. And that paragraph has information about the scientist. Trust me, multiple students ignore that, but that is so much important information. Right now, I can show you. If I'm opening this NCERT, let's say I'm opening biotechnology because it's itself is having 2 chapters, right? I'm opening this NCERT. If you see now, they have given this paragraph. They have talked about a scientist and some information is given. It is there in every starting unit. You see, I have taught this in the class. For sure a question can come from there also. So that's why I'm saying. Hanji. So this is the, I hope you understood this is important topic. Restriction enzyme, cloning vector, transformation process. And other than that, na, just in transformation process, there are multiple topics. For example, PCR, polymerase chain reaction. There is electrophoresis. That is also important. And even there is the transfection wala topic, so much important. That how we are taking transgene, which is the gene of interest, and how we're putting it the host, and by which method we are putting it into the host. So this is what it is. Next is biotechnology and its application. Here you will be learning about BT cotton. Actually, it's not about the BT cotton. It is about all the product. RNA interference, BT cotton, and then there is flavor saver, golden rice, whatever topics are there, which are modified version of the latest variety, how it is done, and what type of genes are there. And like just say BT cotton, they have talked about the cry gene. Cry A1, A, C, and AB, whatever genes are there, it is mentioned in the NCERT. Important, guys, you need to learn that and how it is acting, like very, very important. Anyways, gene therapy there is a history of gene therapy where they talk about SCID. You remember in SCID there is ADA, adenosine deaminase deficiency wala topic. I am putting 5 star on this one. Again, important. You already are having important topic in your plate and you know that I'm telling you. So the thing that you have to do is do it. The first thing. After that, organism and population. Guys, just give me 1 second because I need to put a reminder. I need to call some people in the class so that the student can be there in the class. Okay, just give me 30 seconds. Okay, I would like to continue. I was just calling some people who were supposed to be there in the session. And yeah, so guys, next is organism and population. Into that, you need to talk about population interaction. There are a lot of phenomena, commensalism, mutualism, and parasitism, amensalism. So all of them are important with examples. Ecosystem is pyramid of number, energy. Yahan pe to I recommend that there is a topic called types of ecological pyramid, right? And when we talk about the shapes, there is inverted, there is upright, then there is spindle. What you have to do is to learn an example of it. Like, what do we call? Grassland ecosystem is what shape? Inverted, upright, or maybe desert ecosystem. So they will give you an ecosystem and they will ask you, and that too from the NCERT. Next, you people have to do biodiversity may say cause, that's easy peasy. Environmental issue is deleted, so let's not talk about it. And yeah, acha, I did not mention any other deleted, na? First chapter, this chapter. I'm so sorry. Reproduction. Acha, nahi, I did not talk about the deleted. Actually, in a reproduction organism chapter itself is deleted. I mentioned that mode of asexual is important. But whatever is a part of the next coming chapter, na, that is because this first chapter is something which is deleted, but mixed information is there. Same information you can find in upcoming chapter. So that's why. Chalo, anyways, I think I have told you enough important topic. And as I mentioned, we will be doing this Hardy-Weinberg. Just a second. Okay. My bad, guys. I was putting more messages in the group. Let's continue. Okay, Aniket has joined back. So yeah, the next thing that we are going to do is Hardy-Weinberg principle ka topic. So let's do it. And till the time, I'll be waiting for one student who was there. Hardy-Weinberg principle. Okay. I was just looking for my personal notes that I bookmarked so that I can use it to upload my numericals already that I saved. Let's move forward. Thank you. Aniket, actually, na, you can take a screenshot, beta. I was talking about the important topic and I already have mentioned it here chapter by chapter. That what are the important topic? You're there, right? And the first thing that you have to do is to do these topic because what happened in the exam that your confidence level is very affected when you don't know the key points. So the first thing that you do is you should learn all the important topic. Okay, you can take a screenshot because now I'm going to talk about Hardy-Weinberg and I'll be uploading some numericals also. Okay, let's start it. Let me remind, the topic is Hardy-Weinberg principle. I'm using two screen, guys, but you know, that's why I'm looking on the other direction. Hardy-Weinberg principle. Imagine there is a world where small populations are there, right? This is what I'm talking in the language of Hardy-Weinberg. He's saying that there is an equilibrium, a hypothetical equilibrium where no mutation is happening. So Hardy-Weinberg principle is nothing. It is Hardy-Weinberg equilibrium where no gene mutation is happening. No gene mutation is happening and alleles are not changing. Plus, this is very hypothetical, which is not possible when we even talk about it. But we are taking it as a reference because we have an ideal situation that, you know, suppose there is a value at that reference. From this value, if any plus minus happen, at least we know that how much percent of changes has happened. You're getting my point? Suppose I'm saying this is a line. This is the reference. And if someone is moving from here to there, like you do that in maths, right? What do we call it? I think there are the coordinates, something like that. I'm very weak in maths, but something like that, right? So it's like you need to have that reference. And from this reference, we just kind of, you know, get to know that this is where we are moving and this is the coordinate on X, Y, and whatever coordinates are there, right? Even Z. So point is that Hardy-Weinberg principle is in bio and it is telling this is the reference. This is ideal situation where no gene mutation is happening, no migration in the animals are happening. Animals are not moving from there to there. And plus that, isn't it weird? Random mutation must occur. No gene mutation can occur, but random mating, my bad, random mating can occur. No gene mutation is happening, but random mating can occur. By chance, individuals are mating. Next, no genetic drift happen. No genetic drift happen. And that is a change. I mean, if no genetic drift happen, that means a chance change in allele frequency. That can happen, but in genetic drift, it is like by chance a population is crashing and then one or two organisms which is surviving out of that is not happening. There are just few chance that can happen. Next is no natural selection is happening. No natural selection is happening. So this is the point. Okay. Now, this Hardy Winberg, actually not one, but there are two people, Hardy Winberg, who are talking about the population. And they are comparing the allele frequency. Allele frequency means, suppose I'm saying that out of all this population, 40% population is having a good height. 30% is, so that is the gene frequency, that this much percent of people are having this frequency. You're getting my point, right? And now the same person has given an equation also, that considering all these factors, this is the equation which is possible, which is p square plus q square plus 2pq is equal to 1. That's mathematical equation, but you know, maths is different in bio. So bio is the only subject where multiplication and division means the same, but yeah, that's what it is. P plus q is equal to 1. Now let's understand what it is. I repeat, gene frequency is the frequency of the gene to be present in a population. So if I'm saying, suppose, first of all, let me tell you what is P here. P is the frequency of dominant gene, frequency of dominant gene. And Q is the frequency of recessive gene. This is very much important again. If I am writing the word, even if I'm writing allele, gene or allele, that means the same. That means P and Q. If I am saying it is P square, then either homozygous or heterozygous term will be used. It will be homozygous allele or heterozygous. Achha, sorry, my bad. It'll be homozygous dominant allele. I will tell you why did I say sorry. Because I said a sentence which was a little wrong. Homozygous recessive. Actually, I told the word heterozygous. Heterozygous is used for PQ, 2PQ. This is heterozygous because one is P and one is Q. Let me tell you that in simple terms. Suppose this is a chromosome. Okay, here capital A is present and here suppose capital A is present. So when both of them are capital A, that is dominant. Suppose I'm saying here small a, then homozygous recessive. And so homozygous, if I'm writing, that means it is a pair. So square, P square, Q square, dominant or allele. That means a pair of gene are present. But if simple P and Q are there, then it will be frequency of dominant gene or allele, but that will be the single one. They are just talking about this as a single. They are not talking about the pair. Got it? This is the numerical, this is the formula that you people have to use to solve a numerical. I hope you understood it. Now let's try solving some numerical. Okay. I have lots of numericals with me right now. But as per your NEET exam, you don't get very difficult example. Wait, I'm just checking my notes. I will copy paste a simple example. Found it. Just a second. Okay, so this is the first numerical. I think I'll be ending the session soon because you people are actually, I was expecting more people from my batch, but most of them have exams, so that's why. But that's okay. You people understood. I'll be solving one example and let's see what do we do further. You can see this content, right? I'm going to increase the font size, then it will be more visible. So if you people can see now, this is a numerical. You have sampled a population in which you know that the percentage of homozygous recessive genotype is 36%. So they are telling the value. Then using this 36%, you have to calculate frequency of AA genotype. First question ka answer. They themselves have given answer. They are asking about actually this is homozygous. That means P square. They themselves are telling that the P square is 36%. Got it? Now the next question is frequency of A. So A will be what? P. They are talking about single gene. So now you have to just underroot it. Underroot it, my bad. Achha, by the way, there's one more thing I want to tell you. If it is 36% now, you have to write it 36 by 100. Then the value will be 0.36. Like out of 100%, this is 36%. But when we say out of 1, it will be 0.36 as by definition. And they are telling you that if, acha, they were talking in the numerical in which, you know, the percent of homozygous. Homozygous recessive, my bad, it will be Q square. I thought they have written dominant. Okay, rest story same. So it will be Q square. So they are asking you Q. So they are saying that the Q square is 0.36. What will be Q? Under root 0.36. Got it? So then Q will be 0.6. So Q value is nothing. It will be 60%. You're getting this right. Now, next question is the frequency of capital A. They are talking about capital A. They are talking about the dominant gene. So this will be P plus Q is equal to 1. Easy peasy. Then you know that the P value is 0.4. So Q will be 1 minus 0. Achha, my bad. Wait. P value, I think I have made a mistake here. We already know Q here. So P plus Q is 0.6. Acha, my head already calculated 0.4 here. It will be 0.4. No? 1 minus 0.6. My head already is telling me this is the answer. So this is what it is. It'll be 40%. Next question. The frequency of the genotype capital A. And this capital A small a also. So technically they are asking, now you guys know the value of P is 40% or you can say 0.4. You know value of Q, which will be 0.6 and Q square also, which is 0.36, right? Now you have to calculate P square. This is what they are asking, plus 2PQ, capital A small a. They are talking about heterozygous one. That's what you have to solve. So the frequency of capital A square here, it will be P square. Right? So you can easily calculate it if there is anyone who want to calculate. It will be 16. Na? So if you people have this value, P square, this is going to be 0.16 or you can say 16%. Correct. And if you want to calculate 2PQ, then you people have to multiply. 2 multiply with 0.4 multiply with 0.6. This will be 0.48. I think you people are clearly understanding this, right? This is what it is. And this is how you have to solve the numerical. Okay, there's one more student who is in the waiting room. Hello, hi. Okay, there's some issue. She's connecting. Okay, I think I'll be continuing. There's one student I was waiting for, Sakina Ali. Hi, Sakina. I think her audio is not connected, but that's okay. I think we have practiced Hardy-Weinberg principle, and if any numerical come here, you will be able to solve. I am going to open-end a question here, and I expect that you're going to try solving it. So this is numerical that I'm copying here. You can take a screenshot, and maybe you can try practicing that. Okay? Try to solve it. It is very, very easy. Not very difficult one. This is the numerical. You can take the screenshot and you can try solving it. Okay. Okay. So we have done Hardy-Weinberg principle. There is one more thing I would just like to upload. I wanted to talk about actually lac operon. So I'll directly upload a picture. Just a second. Hello. Sakina, am I audible, beta? Could you please confirm? Okay, so basically, Sakina, I have been taking session. Two students were getting online and offline, but since we are live here and I'm just discussing some topic, let me repeat it. Till now, I have talked about all the important questions, all the important topic, you can say, which is important for your board exam. So I have highlighted it all. It'll be nice if you want to take a screenshot. After that, I was talking to the other student in the class and I was asking the Hardy-Weinberg principle, and he told that, ma'am, we have not covered the numerical. So I was talking about Hardy-Weinberg principle. I talked about it and I gave formula, and one numerical also I have solved it. Got it? Live session will be available on YouTube. You can back it and you can watch it. This numerical wala part, because this topic is very important. There are 90% chance that you can get a question from this topic. Okay, now the next topic that I was thinking in my head that I wanted to revise with you is lac operon. Do you know lac operon? Just confirm me, because I actually copied it not clearly. Oh my God. Okay, I'll try to simplify it, because let's continue. What else do I say? So basically, dear, lac operon is it is like, first of all, let me tell you what is operon. Operon is an organization. Like, suppose I am saying that a chromosome is having a gene, and that gene is expressing itself, and that gene is making a protein. Suppose I'm saying that there is a gene which is controlling my height, that is making growth hormone, but that gene is not a concrete unit. It is an organization. It is like askIITian is teaching you, but askIITian doesn't have teachers, right? There is support team also which is supporting. There is IT people also who are maintaining website. There is mm, teachers, and then there are people who are making your question papers. So there are different teams who are working, but all you know are the teachers, right? That, okay, I have a class, but you don't know who is making the website, who is updating the link, this and that, right? Similar way, a gene is there. If I am saying, suppose this is the chromosome and this is a dot here, which is nothing but a gene which is controlling, but that gene is a structural unit. This gene is made up of so many things, which we call it as operon. Operon is a model. It is made up of units which are doing different work. It is having regulatory gene. It is having promoter gene, promoter area. You're getting my point? There are different areas present on it. It is having regulator, promoter, then it is having operator, which is operator, but yeah, in this case blocked. I'll talk about it after that. Structural gene. These structural gene are the one which are actually working. You're getting my point? These structural gene are the one which are making the product. If I'm saying in case of lac operon, there are three structural gene present: lac Z, Y, and A. They are, and this is there in the NCERT. Lac Z, they are making the product. They are making the actual product. You're getting my point? There are two cases. This picture is explaining one and two cases. In first case, the structural genes are turned off. They are not working. It's holiday. askIITian doesn't have any class. It's gadgeted holiday. Maybe it is Diwali or any other season. Got it? But the other one is when it is working. It is Monday, or maybe it is Sunday also, right? You're getting my point? So at this point of time, second time, properly genes are active and there is a product present. But how it happened? So in first case, what happened? There is a repressor present, and that repressor is blocking something. You remember when DNA is making product, DNA make RNA first. That DNA is double stranded. Some enzyme would come. That enzyme will be attached here and it will start making RNA out of it. Right? But in first case, you see that operator is being blocked. Actually, there is this enzyme present, which is RNA polymerase, and it is blocked. It cannot move further. Understood? And that's why structural genes are not working. But in second case, regulatory gene is making a product which is releasing repressor, but that repressor itself is repressed. It is lack operon, so there will be lactose present. That lactose will go attached with the repressor and block it. Repressor cannot work. RNA polymerase will slide over and do the work. Sakina, are you able to understand the story I just told you? Good girl. Now let me tell you the important thing. LAC-Z is, okay, I'm using a bad color again. LAC-Z is coding for galactosidase, beta galactosidase. It breaks the galactose actually. Can you repeat? I am telling that there are 3 structural genes present. LAC-Z, Y and A. And these genes are making different product. So dekho, let me tell you that whenever, not from starting. Yeah, but I want to tell you one thing. Like what happened now? In our body, whenever DNA want to speak, it talk in the language of protein. It would make a protein and then talk. In this case, as they have written Z, Y, and A, they are also making protein. Galactosidase is an enzyme, but that is protein. Enzymes are made up of protein. So Z is coding for beta galactosidase. Then second, Y and A. This Y is coding for, this is acetylase, trans acetylase. Permease, my bad. Permease enzyme. Because actually what happened, na, जैसे मान लो imagine it is a cell. Actually, permease enzyme na affect the permeability of the membrane so that lactose can move inside. Because obviously it is lack operon, it need lactose to work. So permease will go and affect the permeability of the membrane so that more lactose and even glucose also can come inside. Got it? Galactosidase break lactose. Galactosidase जो है ना, I'm sure you have learned that already. It break lactose into glucose and galactose. Glucose and galactose. Okay, I have told you the short story, but you understood it. Now tell me what is your doubt. I can repeat it again, but you want me to repeat it all? No, ma'am, it's clear. Pakka na, matlab I tried the shortest way possible to tell you. Now think about 30 seconds, process the information and tell me the next doubt. Because I'm all vela to tell you if you have any doubt. Please make use of it. And this diagram is like really important and really nice. This is my favorite diagram. This diagram is expressing everything which is possible. Okay, so I'll keep my camera on, but it is clear. I'm just giving you a time. If you have any doubt, ask it. Okay. Hanji. Any doubt? No doubt. Pakka. Chalo, theek hai. So I am going to give you, then I will be ending my session, but I want you to see this YouTube wala because this Hardy-Weinberg principle is important. I want you to see this part because I was explaining how you have to solve it. Now look at this. These are important topics from all of the syllabus. Wherever you think there is doubt, I'll take it. You're free to decide. I'm zooming it. Just a second. Good luck to you, Vita, for your exam. Yeah, ma'am, can you explain biodiversity loss? Biodiversity loss. That's very important topic you're asking. So basically, from biodiversity, how biodiversity loss happen and what are the reasons? That's what you have to learn. So, okay, let's move. Acha, by the way, I can do one thing. My notes are already there. I can refer to that. It will save time since we are short of time now. Here, class notes. Biodiversity. I'm saying, where are my class notes? I was scared for a while. Why have I even changed the color? Acha, there might be reason. Okay, Amazon species area importance. Loss of biodiversity. Okay. Actually, yeah, एक चीज. From the loss of biodiversity, na, in your NCERT, some numbers are explained. I actually taught from the NCERT because you have to learn as it is number. Trust me, because जैसे क्या होता है, जैसे नंबर में ना they give year according to data 2020, according to data 2012. Your NCERT doesn't have very latest color information, but whatever is there that you have to remember. जैसे यहाँ पे I talked about that IUCN, it is International Union of Nature and Natural Resources. In 1964, they have, this was actually originated and they have started to collect all the data. It is like they're having data. IUCN organization is having data of all the animals. If you check website, it is there. All the animals which are extinct, which are threatened, which are vulnerable. There are multiple categories we know. So according to their book, which is 2004, this is what it is there in the NCERT, 2004 data, 338 vertebrates, 359 invertebrate and 87 plant, they are like red data list. Extinct 784 species in last 500 years. They have vanished. After that, there is, I was actually reading this from the NCERT, I remember. I might have highlighted also. You have to spend some time in remembering those number. I'll be very honest. You see, I have told this in the class that this animal which is there, like you see that the colonization of tropical Pacific island by human is said to have led to the extinction of more than 2,000 species of native birds. You need to remember that, beta, because biodiversity loss, you know, by how biodiversity loss happen. There can be invasion, there can be any drastic cases, there can be obviously human for multiple reasons, but you have to remember the animal's name. As you see, at present, whatever I have highlighted, they're important. As you see, dodo in Mauritius, quagga in Africa, thylacine in Australia, stellar sea cow in Russia, and 3 subspecies in Bali, Javan, and Caspian of tiger, they're gone, extinct. Then, the last 20 years alone have witnessed disappearance of 27 species. You are not able to actually remember it. Are you able to like having problem in understanding or remembering it? Because it is all fact. As you see, adding to the grim scenario of extinction, more than 15,500 species of worldwide are facing threat of extinction. Presently, 12% of all bird species and 23% of mammal species, 31% of gymnosperm, threat of extinction. So this paragraph is just threat, not extinct, but threat. You're understanding it? You will have to move forward, okay? Okay. So now here you see how is the sixth extinction present in progress different from the previous episode? Oh, I think I saw a question in the pre-board. Some student shared that with me and it was there. That how it happened. The difference in the rates, the current species extinction are estimated to be 100 to 1,000 times faster than in pre-human. You know the reason. So, causes of biodiversity may habitat loss and fragmentation. And see, in short, in your NCERT, they have talked about how biodiversity loss happen, what are the reason, and then they have given fact. If you have, dear, any question in the paper, I request that you write the fact and you learn it. As soon as you write the fact, full marks. Because as a biology teacher, now I love reading about example, real life example and facts. You write the fact and underline it. Now, there is a fact that 14% of the Earth's land surface is having tropical rainforest, which now covers 6%. It is a fact. इससे clearly पता चलता है that the student know. You're getting my point. So other than just writing vague lines and everything, general, and please learn fact here. Make quick notes. Do one thing, go through the NCERT because this chapter is full of that and write the fact. Got it? So that will be my suggestion. So dear, I think I'm done with today's lecture. And if you have any doubt or something like that, please reach the team. You know that support at the rate askIITian ID is there. And if you have any concern, let us know. I can take one more session and just take the doubt removal session. Okay. So thank you so much, guys. Thank you for watching the video and thank you for being live with me. And some student of mine have their session, but I decided that I'll keep on the session going. And I will tell you the important topic. I talked about two topics, which is lack of operon and Hardy-Weinberg principle, and little about biodiversity loss. But I request that you reach the team. And if you have any doubt, we will take it. Thank you so much. Bye bye. Good night. Take care.